Hello everyone. Today we are going to discuss about Corynebacterium. Corynebacterium diphtheria is a pathogen that causes diphtheria. So in this context, we are going to discuss about or our objective is to study its morphological and cultural characteristics, understand the pathogenesis, mainly toxin, clinical manifestations of diphtheria, lab diagnosis of diphtheria, prophylaxis of diphtheria. We all know diphtheria is caused by Corynebacterium diphtheriae. This Corynebacterium diphtheriae is otherwise called Klebsiella bacillus. This is an aerobic, gram-positive, non-acid-fast, non-sporing, non-motile, and pleomorphic organism. These are thin, slender, irregularly stained, club-shaped, gram-positive bacilli showing the presence of volutin granules at one or more ends. On staining with Loeffler's methylene blue, the granules of this organism take up bluish purple color, and these granules are called metachromatic granules. So there are other granules like Babes and granule, which is composed of polymetaphosphate and act as storage granules. These bacilli are seen at angular fashion resembling V or L shape. So we call that arrangement as Chinese ledger or cuneiform arrangement. Staining methods for the detection of Corynebacterium diphtheriae include Albert stain, Ponder stain. And the principal virulence factor of diphtheria is the toxin, diphtheria toxin. Coming on to cultural characteristics of Corynebacterium diphtheriae, this organism grows best on media enriched with blood, serum, egg, etc. These are aerobic, facultative, anaerobic, and grows at an optimum temperature of 37 degrees Celsius and pH 7.2. Media used for the cultivation of Corynebacterium diphtheriae are his serum water, it's a liquid medium, Loeffler's serum slop, in that they appear as small, circular, white, creamy, glistening colonies. Then tellurate blood agar medium. This is a selective medium for growing this Corynebacterium diphtheriae. That is tellurate blood agar medium. And thus, potassium tellurate is a selective agent, and they appear as gray to black colonies due to the reduction of potassium tellurite to tellurium. There are three biotypes of Corynebacterium diphtheriae in this medium, that is Corynebacterium diphtheriae gravis, which shows daisy head colony, Corynebacterium diphtheriae intermediate, which shows frog's egg colony, Corynebacterium diphtheriae mitis causes poached egg colonies. This bacteria ferments glucose and lactose with acid production. They are sensitive to heat, destroyed by antiseptics, and remains virulent in blankets and dust for about 5 weeks. This is the figure showing the growth of Corynebacterium diphtheriae in Mitis chocolate tellurate agar. So next we will discuss who are the reservoirs of this diphtheria, or who all are the reservoirs behind this diphtheria infection. So the reservoirs are human carriers, usually asymptomatic. Transmission is mainly by respiratory or rarely through skin and fomites. The temporal pattern responsible for this infection is during winter and spring. Coming on to the pathogenesis, we already said that the virulence factor behind this diphtheria is diphtheria toxin. That is, pathogenicity is due to powerful exotoxin. Toxicity is due to the presence of toxin transferred from one bacterium to another by beta-phage. This exotoxin is a protein of molecular weight 62,000 Dalton and have two fragments A and B. In this, fragment A has enzymatic activity and fragment B is responsible for binding and transport of toxin into cells. The toxin is heat labile, extremely toxic and capable of producing antitoxin. So coming on to the mechanism of action of diphtheria toxin, which is very important in pathogenesis. This toxin acts by inhibiting protein synthesis. How this happens? Fragment A of this toxin inhibits polypeptide chain elongation in presence of NAD by inactivating elongation factor 2, thereby inhibition of translation takes place. This toxin has affinity for myocardium, nerve endings, and adrenals. Toxin production is influenced by concentration of iron, that is 0.1 mg. This toxin is susceptible to penicillin, erythromycin, and broad-spectrum antibiotics. This is the figure showing the mode of action of this diphtheria toxin. A has enzymatic activity. B will bind to the host cell receptors. Factor A, which will inactivate elongation factor 2, thereby prevents protein synthesis by ribosome and causes cell death. So next, coming on to the pathogenesis, mode of transmission. Main mode of transmission is through contact or through droplets of secretion. And the portal of entry is through respiratory tract, or the organism may enter through conjunctiva or skin wound. These organisms also inhibit mucous membrane. Upon entry, the bacilli multiply locally in the throat, produces powerful exotoxin, and produce local and systemic symptoms. Most commonly affected site of diphtheria is the phoceal site. Diphtheria can be classified based on site of infection as respiratory or phoceal, which is the commonest type, nasal, laryngeal, ocular, genital, cutaneous, which will be presented as punched out ulcerative lesion with necrosis, most commonly in extremities. Then systemic complications include polyneuropathy and myocarditis, pneumonia, renal failure, late toxic manifestations after weeks of infection. So up to this, we had discussed about Corynebacterium diphtheriae, their morphology. After that, we had discussed the virulence factor or pathogenesis of this diphtheria. Later, we came to the pathogenesis, that is mode of transmission, portal of entry, and the classification of diphtheria. So next is local lesions during respiratory diphtheria. The exotoxin elicits an inflammatory response which causes the necrosis of epithelial cells, which will liberate serous and fibrinous exudate leukocytes, erythrocytes, and bacteria. Mucosal ulcers results lined by a grayish-white pseudo-membrane, and this membrane appears on tonsils and spread to larynx and trachea, causing fatal airway obstruction. Occasionally, this membrane may be formed on uvula, soft palate, gingiva, buccal mucosa, and site of tooth eruption. Membrane bleeds on being dislodged, and the surrounding tissue is inflamed and edematous. So these are the systemic complications like polyneuropathy, myocarditis, renal failure, pneumonia, then toxemia. They can act on cells of cardiac muscles, adrenals, and nerve endings. We already said that this toxin has affinity towards cardiac muscles, adrenals, and nerve endings. Next, we will move on to clinical manifestations. The major clinical manifestations are high-grade fever, pain in throat, ulceration of membrane followed by cellulitis or gangrene of surrounding area, hemorrhages at the edge of membrane, cervical adenopathy in severe cases, and death is mainly due to circulatory failure or mechanical asphyxiation. Depending on the severity, there are three types of diphtheria, that is malignant. First one is malignant, that shows severe toxemia, cervical lymphadenitis, massive tonsillar swelling, and neck edema. That is bull neck appearance, circulatory failure, high incidence of paralytic complications. So this is what we seen in this picture. Next complication or next severity is septic, which leads to ulceration, cellulitis, and gangrene around the pseudomembrane. And the last severity type is hemorrhagic, characterized by bleeding from the edge of membrane, epistaxis, conjunctival hemorrhage, and purpura. So moving on to complications, asphyxia due to mechanical obstruction takes place, acute circulatory failure, post-diphtheritic paralysis, and septic complications such as pneumonia and otitis media may occur. Next is laboratory diagnosis. So after clinical manifestations, we are going to discuss about laboratory diagnosis. First one is the collection of specimen. Or the site of specimen is swabs from lesions, throat, nose, larynx, ear. 2 swabs are collected, one for smear and one for culture. The next one is direct microscopy by gram staining, gram-positive bacilli. Then we already said demonstration is by Albert stain. Beaded green rods, Chinese letter arrangement we can observe. Then culture on blood agar, low fluorescence serum slow, blood potassium tellurate agar. Incubation period is for 37 degrees Celsius for 48 hours. Growth is confirmed by microscopic examination of colony smear, stained by Albert stain. Metachromatic granules were seen and cuneiform patterns were visible. Biochemical reactions like they ferment glucose, maltose with acid, no gas, they are catalase positive, reduces nitrate to nitrite. So the next test under lab diagnosis, virulence test. There are 2 types of virulence test, in vivo test and in vitro test. In vivo test can be done intradermally or subcutaneously. So subcutaneous test, how we can done, that is growth from overnight culture on low fluorescence serum slow is emulsified in 2 to 5 ml broth and 0.8 ml of emulsion is injected subcutaneously into 2 guinea pigs. One received intramuscular injection of 500 units of diphtheria antitoxin 18 to 24 hours previously, and the unprotected animal dies within 2 to 3 days with evidence of hemorrhage in adrenal glands. So autopsy of that animal shows hemorrhagic edema, necrosis at site of inoculation, swollen and congested lymph node, congested abdominal viscera, peritoneal and pleural exudate. The next test is intracutaneous test. 2 guinea pigs injected intracutaneously with 0.1 ml emulsion from growth on low fluorescence serum slow. 1 animal protected with 500 units of antitoxin intraperitoneally 4 hours after skin test in order to prevent death. Other animal is given 50 units antitoxin intraperitoneally 4 hours after skin test in order to prevent death. For toxigenic strains, inflammatory reaction takes place at the site of injection, progress to necrosis in 48 to 72 hours in test animal. Advantage of this intracutaneous test is that 8 to 10 strains can be tested at a time on a pair of animals and animals do not die. The next important test for the diagnosis is in vitro test, and in that LX gel precipitation test. It's an immunodiffusion test. A rectangular strip of filter paper soaked in diphtheria antitoxin is placed on the surface of 20% porous serum agar plate in molten condition. After solidification, test strain is streaked at right angle to filter paper strip with or without controls. Plate incubated at 37 degrees Celsius for 24 to 48 hours. The toxin diffuses in agar and produces a line of precipitation where it meets with antitoxin at optimum concentration. Non-toxigenic strain, there will be no precipitation line. This is about LX gel precipitation test. This is an in vitro test used, it's a virulence test used in lab diagnosis. So up to this, we had discussed the morphology of diphtheria, pathogenesis of diphtheria. Then we had discussed what is the mode of entry, what are the types of diphtheria, severity types of diphtheria. After that, we had discussed clinical manifestations of diphtheria, then lab diagnosis of diphtheria, and the last but not the least, we are going to discuss about prophylaxis. There are 3 methods for prophylaxis. The first one is active immunization. At 6 weeks of age by toxoid in combination with tetanus toxoid and pertussis vaccine, DPT. 3 doses intramuscularly 4 to 6 weeks intervals. Booster dose can be given 18 months and 5 years. Then coming on to passive immunization, emergency measure for susceptible people exposed to infection, 500 to 1000 units anti-diphtheritic serum ADS is given subcutaneously. Then combined immunization was for person receiving ADS plus alum precipitated toxoid is preferred. That is anti-diphtheritic serum plus alum precipitated toxoid is preferred in combined immunization. Then Schick test, that is toxin antitoxin neutralization test, that is intradermal test to demonstrate circulating diphtheria antitoxin. That is 0.1 ml diphtheria toxin injected intradermally on one forearm, same amount of inactivated toxin injected on other forearm. Results rate after 1, 4, 7 days. Positive reaction shows area of swelling and erythema at the site of injection of toxin after 24 to 48 hours, reaching maximum of 4 to 7 days and measures 1 to 5 centimeter and then fading away. Control and absence of any reaction, that is a negative reaction, indicates toxin neutralized by sufficient amount of antitoxin in blood. Erythema in both forearms within 6 to 24 hours and disappears within 4 days, that is immune and hypersensitive to components of diphtheria bacilli. That's a pseudo reaction. Then the last but not the least, there's a treatment that is sensitive to penicillin erythromycin but of little value. In suspected cases, anti-diphtheritic serum should be immediately given. So this is all about diphtheria. So diphtheria is caused by Corynebacterium diphtheriae. Morphology, that's a gram-positive bacilli, shows the presence of pollutin granules, metachromatic granules. Then their cultural characteristics we had discussed. Main mode of pathogenesis is through toxin, diphtheria toxin, and the mechanism of action of diphtheria toxin we had discussed. Then in pathogenesis, mode of entry, then who are the reservoirs, types of diphtheria we had discussed. Then we had discussed about the lab diagnosis of diphtheria. Then next one is treatment, prophylaxis and treatment of diphtheria. So this is all about diphtheria. Thank you so much.